Good afternoon to everyone. We'll be starting our session shortly. Good afternoon, sir. Good afternoon. You can unmute, sir. Actually, I made you co-host. You can unmute yourself and you can speak. There is an unmute. So you can unmute your mic and you can speak.
Yeah. Is it okay now? Am I audible? Hello.
Yeah. Yeah, it's okay. Yeah. Our team are here in this room. Dr. Bhargav, organizing secretary, also there. I'm in charge for this IDP Nahi project.
Okay.
Welcome you, sir, for this afternoon.
Thank you.
As a resource person.
Yeah.
Dear participant, this is our last session of our 2 days webinar on this nursery production and management of horticulture crops for young entrepreneur. So today our esteemed resource person, Dr. D. Prasad, Principal Scientist, Division of Crop Improvement, CAR, IISR, Record. And I would like to read his biodata briefly. Dr. D. Prasad is presently working as a Principal Scientist the ICR Indian Institute of Spice Research, Kozhikode. He did his postdoctoral research at University of Gulf, Canada, under the prestigious Weizsäcker Fellowship during 2009 and 10. He's also recipient of Australian Government Entrepreneur Fellowship during 2017 at University of Queensland, Brisbane, Australia. His focus area of research is improvement of horticulture crops through conventional and genomic approaches. He has 20 years of research experience in horticulture crops and made significant contribution in the field of spice, genetic resources, and breeding. He was involved in the release of 5 high-yielding varieties in spice, also standardized technologies for quality planting material production in ginger and black pepper. He has identified basal defense mechanism against bacterial wilt resistance in ginger through mRNA sequencing, which would be helpful in developing wilt resistant varieties. He has published over 50 peer-reviewed research articles in various national and international journals. He is recipient of J.S. Fruit Award for the best research paper, National Academy of Biological Science and Best Research Paper Award 2013, H.S. Mehra Memorial Award for Best Research Paper 2015, D.P. Ghosh Young Scientist Award, Horticulture Society of India 2017, ICR Faruddin Ali Ahmed Award 2019. He's a fellow NARS, IAHS, and ISS. Dear participant, it's a we can have it this topic at this end, but then it is more important for us production of quality planting material, particularly for the spice crops. It's very much important in Northeast. If you see that Northeast have rich in spices, and we have a lot of scope, tapping the resources and how to multiply and supply the planting material. So here with us, Dr. D. Prasad, sir, principal scientist, will be more focusing on this topic. And the topic is quality planting material production in spice crops. So it will be a wonderful opportunity for all of you to interact with him at the end. If you have any set of question, write it on the chat box, or else you can interact with him at the end. Sir, now it's a time, sir, please take a time and start your lecture.
Yeah. Thanks, Dr. Raja, for nice introduction. And also Dr. Bhargav for contacting me and arranging this lecture. So, yes, Dr. Raja said the quality planting material plays an important role, especially for spices in northeastern region. So I am planning my lecture two modules. Maybe the first 45 minutes, 45 or 50 minutes, like I'll be highlighting the importance of quality planting material in ginger. So because that is the important crop in Northeast. So my first lecture is on ginger and turmeric, mostly the planting material production and some of the important varieties. So then second lecture on quality planting material in black pepper. Because these three are the our target crops as far as government of India is concerned to the northeast. So I'll be concentrating on these three crops. So if it is okay, so I can go ahead with my presentation.
Yes, sir, you can.
Yeah. What is the composition of participants? So can I have a brief idea about who are all the participants?
Yeah, sure, sure. Actually, more than 350 students are registered for this webinar,
Okay.
and some of them are in now in Zoom platform, and many of
Okay.
them watching on offline YouTube. So
Oh,
if
okay.
they have a network issues, they can watch later on also. It is streamed on the live on YouTube. We will be
Okay.
sending the link to the participant. They can view. And as
Okay.
of now in YouTube also, many people are watching and they are
Okay.
participating in Zoom platform.
Oh, nice.
Mm.
So, uh, like, so we'll start with the ginger presentation.
Sir, you can click the share screen. Oh, yeah, it has come. Yeah,
It has come now. Yeah.
yeah, it has come.
Yeah. Yeah. So as I said, so this lecture is about quality planting material production and varietal status in ginger. Because why I'm clubbing varieties into this lecture is, so without varieties, like talking about the quality planting material is not fair. So varieties plays an important role. So then like the quality planting material. So these two go hand in hand. So we are from Indian Institute of Spices Research. So you're all aware that it is a ICR institute located in Kerala. So our mandate is spices. So we work on black pepper, cardamom, ginger, turmeric, vanilla, paprika, and all tree spices like cinnamon, garcinia, nutmeg, clove. Uh, and besides that, ICR has one more institute for seed spices in Rajasthan. So that is for NRC on seed spices. So we work mostly on major spices. So am I audible to all?
Yes, sir, it's audible. You can continue.
Okay, thank you. So coming to the ginger production, if you look at the global the production status, so the globally you can see the map. So where ginger is cultivated across the tropical countries, and the total area is 3 lakh 73 hectares, and with the production of almost 28 lakh tons. So this is the global production. And if you look at the top 10 ginger producing countries, so India stands first, followed by China, Nigeria, Nepal, and Southeast Asian nations. So these are all the top 10 ginger producing countries. And if you look at almost 82% is produced in Asia, and Africa, followed by Africa with 17%. So these are all the major ginger producers by region wise. If you look at the just a few slides about the ginger area and production, so that you will come to know the importance of ginger in both at the national and the global level. So if you can see the export of ginger is like 885 million US dollars. So worldwide. So and China stands first with 471 million US dollars. Though we are the major producers, China has the maximum export of gingers, which earns 471 million US dollars. Then next is Netherlands with 100 million US dollars. So they export ginger worth 100 million tons. Then like we have only 31 million US dollars worth exports from India. And so other side, if you see, like USA is the major importer, followed by Japan, Netherlands, Pakistan, and Germany are the major importers of ginger. And if you look at the commodity wise, so ginger, fresh ginger constitute 48% of the export, then 44% dry, then rest all ginger powder, oil, and oleoresin. Coming to the Indian production, like so our area is around 1,70,000 hectares with 18 lakh tons of the production. So then you can see as on today 2018-19 area. So Madhya Pradesh stands first with the maximum area, followed by Karnataka, West Bengal, and Odisha. So you can see the productivity also. So Madhya Pradesh has the maximum productivity and of 16.7 tons, and Odisha with 7.72 tons. Coming to the northeast, why ginger is important, you can see like almost 30-35% of the ginger area is like constituted by northeastern regions. So Sikkim and Assam. So Assam has around 20,000 hectares. So Sikkim has 15,000 hectares. So, and also the other problem you see compared to other states, the productivity in Northeast is comparatively less. You can see like 5 tons, 6 tons. So it's hovering around like 5 to 7 tons per hectare. So whereas the rest of the country, the productivity is slightly better. So in this particular scenario, the planting material plays a major role. And if you look at the ginger cultivation in India, so it is a very cultivated in diverse climatic conditions from tropical to subtropical, whereas down south and central India, it is cultivated in the tropical areas. In the northeast, like it is subtropical to temperate conditions, ginger is grown from plains to 2,000-meter elevation. Then, like especially in Kerala, Northeast, it is cultivated as a rainfed, whereas in Karnataka, Madhya Pradesh, Maharashtra, it is cultivated as a irrigated crop. Then also the like it is cultivated as organic in Northeast in some of the states, especially in Sikkim. And now the people are cultivating ginger under protected cultivation in Gujarat and Maharashtra. So coming to the varietal status, I briefly touch upon the important varieties. So the genetic diversity of ginger is very high in India, especially you can see the high genetic diversity in Northeast and Kerala. And you can see the variability for rhizome characters. So you can see slender rhizomes, bold rhizomes, and like even for quality, like high essential oil types, low essential oil types. Even if you cut a ginger and open and see, So you can see white types, light yellow types, blue types. So the extent of variability is very high for ginger in India. And besides that, we also have red ginger types cultivated in Kerala and Northeast. And also we have the black ginger, which is not in the genus of gingiber. It is a Camphilia parviflora, but it is called a black ginger. It is a highly medicinal. So, what under Indian Council of Agriculture Research and the National Agriculture Research System, so we have like 1,200 accessions of ginger collected from ginger growing regions and cultivate and conserved in our germplasm repository. And if you look at the cultivar status, local cultivars, and you can see like Andhra Pradesh, so they have a local type. So Arunachal Pradesh, you might have heard about Keki and some of the Silang types, Assam. Even in like Northeast, you might have heard about Gurbatani, Baise, Nadia. So these are all the some of the local types which are popularly cultivated across ginger growing areas. And if you look at the characters of some of the local gingers. So you can see the maturity is around 200 to 20 days, and dry recovery around 20%. Root fiber, which is one of the important parameter. So it ranges from like 3 to 8%. So when fiber is less, if it is less than 5%, like it is suitable for processing industries and also for vegetable ginger type. And another important parameter is essential oil. So generally Indian gingers, so which has around 1.5% essential oil. But now some of the northeast gingers like red ginger, which has around 3-4% essential oil, which are suitable for essential oil extraction industry. And you all know this is the popular cultivar in northeast Assam and West Bengal, Nadia, the very the bold type, extra bold type. And coming to the down south, so here the popular local cultivar is Rio-de Janeiro. So this is and you can see the system of intensive cultivation which is followed in Karnataka. And this is how the rhizome development takes place, starting from the planting to 6 months. And you can also see as I talked about the variability for rhizome and the quality characters. And you can also see some of the genotypes like Moran, so which is susceptible to leaf spot diseases, whereas Rio de Janeiro is completely free from leaf spot diseases. So both was planted on the same day in the adjacent field. And coming to the improved varieties, so number of varieties are released from different research organizations. So from Indian Institute of Spices Research, we have released three important varieties like IASR, Varada, Mahima, and Rajeta. And the one more organization from Orissa Agriculture University. So they have a high altitude research station in Potangi in Orissa. So they have released Suprabha, Surichi, Suravi, and Subada. So these are important varieties suitable for Orissa. And besides that, YS Farmer University of Horticulture and Forestry, they have released a variety called Himagri. And uh, Kerala Agriculture University, they have released three important varieties, Adira, Kartika, and Ashwati, which has a high essential oil of around 3%. So this is the variety released from Indian Institute of Spices Research, IASR Varada. You can see the very high yielding variety with medium bold rhizomes. You can see the like a field view of IASR cultivated in Karnataka, and you can see the rhizome characters of IASR Varada, which is suitable for fresh ginger type. And so another variety is IASR Regeta with bold rhizomes, and another variety Mahima, which is quite popular in Karnataka. And if you look at the yield level of some of the ginger varieties, like being a rhizomatous crop, so the yield level varies considerably depending upon the cultivation system you follow. So, for example, Varada, you can see it ranges from 16 tonnes to 43 tonnes. So, so under intensive cultivation system, even 43 uh, uh, tonnes per hectare is possible. But even you can see Rio de Janeiro, even people are harvesting up to 80, 85 tonnes per hectare. And so these are all some of the varieties released from Kerala Agriculture University, Adira, Kartika. The important features of these varieties from Kerala Agriculture University are they have high essential oil content. So generally, as I said earlier, the Indian gingers have like 1 to 1.5% essential oil. So these varieties they have around 3 to 4%. So which are suitable for essential oil extraction industries. So this is the another variety with high essential oil. And so this is one of the recent variety released from YS Parmar University. So the variety called Griganga. So with high recovery of 21%, 1.5% essential oil. And the UBKV, Uttar Banga Krishi Vishwavidyalaya, Pundiwari, West Bengal. So they have also recently released a variety called Mohini, so which is suitable for West Bengal and Assam. So this is what I said. So we have some collaborative project with the northeastern region, and we have collected and characterized some of the important local types in northeast, and we have identified this particular type that is northeastern region one, which has 4.3% essential oil, and you can see fiber content is around 10%, so which is suitable for dry ginger. Besides that, uh, um, the main the pungent principle in ginger is because of gingerol and shogal. So these two are the indicators of good ginger. So you can see these two varieties with almost 2% gingerol and very significantly high shogal content. So compared to, you can see this table, so where we compared with some of the popular cultivars of Nadia, Rio de Janeiro, and Baise. So these two types are significantly better for essential oil and suitable for essential oil industry. And you can see the dry recovery, especially that northeastern region one has 25% dry recovery compared to some of the local types. And another important factor, especially for the quality of ginger, is genotype environment interaction, what we call as G into E interaction. So, because the northeast ginger, which has very high gingerol content, the moment you bring and cultivate in down south, so the gingerol content will be very low. So that is called the genotype environment interaction. So we have conducted a trial across our All India Coordinated Research Program on spices, and we have identified some of the varieties as stable varieties. It means like even if you cultivate in northeast or down south or Maharashtra, Gujarat, so you will get the stable the quality parameters. So another important area is geographical indication. So as far as ginger is concerned, like you may be aware that so this is Kirby Anglong ginger, so which got GA registration. So mostly cultivated in Kirby Anglong area, and and it is organically grown, and so they have a GinFed, Ginger Growers Cooperative Marketing Federation. So they are doing successfully, and it has a huge export potential. And so this is the another GI, not in India, but in Malaysia. So especially that Bentong ginger they called. So Bukit Tinggi area of Malaysia. So they got the GI, and so they have the huge export potential for this ginger. So especially, so why I am emphasizing geographical indication is, so the entire Northeast India, so we have to identify some of the potential GIs, and we need to promote the GIs so that it can be fully utilized. So coming to the world ginger cultivars, and China, Jamaica, Japan, Malaysia. So these are some of the countries they grow ginger, and so they have number of cultivars. And this is the Chinese gingers. You can see it is like our Nadia, uh, very bold, but fiber content is very less, and it is mostly used as vegetable ginger. And Australia, so they have a small pocket for ginger. So, and they have three different cultivars called Canton, Queensland, and Jamaica. The Canton very bold vegetable types, and the Jamaica is a slender, uh, high pungent types. Queensland is in between and used for processing industries. And also they have developed a variety called Buttering Gold, so which is a tetraploid. So ginger is generally a diploid, 2n is equal to 22. So through polyploid breeding, so they have developed a tetraploid with bold rhizomes suitable for processing industry. And these are all the some of the ginger types available in Indonesia, like red, yellow, and white types. So as we talked about varieties, so the next question is whether the governmental organizations will be able to supply the quality planting material. Like it is a big question mark. So to overcome that, so ICR has introduced a policy called varietal licensing contract. So wherein IAS are varieties. So entrepreneurs, because I heard from organizers that some of besides students there are many young entrepreneurs also attending this training, so this information may be useful for them. So this is the ICR licensing policy of varieties, wherein entrepreneurs can take a license from ICR institutions. So, wherein ICR Institute will be visiting your farm, and so we will take care of the quality of the planting material. So we will be visiting and certifying that there is no mix-up, there is no disease in the field. So then, like after harvesting, so the entrepreneurs can distribute the planting material in the name of ICR varieties. So this is the system we are following for spices varieties for past almost 10 years. And it is like an exclusive licensing system. So you can take a license and anybody approaches us. So we will be giving the license to them. So our main objective is to distribute the quality planting material. And so for that, like you have to enter into the agreement with ICR institutes. And it is initially for the 3-year period. And so we will be visiting the field to see the growing conditions and the mix up and other aspects. And finally the ICR logo and ISR logo can be used by the entrepreneurs to distribute the planting material. And you can see last 10 years, so we have 27 licenses. So we have given across the countries. So so especially for turmeric, ginger, and black pepper varieties. So coming to the second part of the first presentation, like quality planting material production. so which plays the very, very important role in ginger, because the two major production constraints in ginger are bacterial wilt and rhizome rot. So both the pathogens like Ralstonia in the case of bacterial wilt, Pythium in the case of rhizome rot. So both are seed borne and soil borne. So quality planting material, especially the disease-free rhizomes, play important role in ginger production. So to overcome that, like we have developed a seed treatment package, and also storage is another important area. Especially ginger is harvested like January, February. Then again, it is planted only by April, May. So it has to be stored for 3-4 months. So that plays a critical role. So the storage condition, ideal temperature should be around 22 degrees, and so it should be stored in well ventilated place. And there are different methods of storage. And we have two seed treatment recommendations. Like what we recommend is like before, after harvest, it has to be treated and stored, and that is the first treatment. The second treatment, before planting, again it has to be treated and planted. So basically, two seed treatments has to be followed. So we have recommendation for both organic and also inorganic. So in the case of inorganic, we have like a chemical. We recommend mancozeb or carbendazim uh, combined with quinalphas. So these chemicals, the seed rhizomes has to be dipped in for 30 minutes. Then it has to be set dried and stored. So this is the pre-storage treatment. In the case of organic, like we have the PGPRs and trichoderma. And this is one of the seed storage method followed in South India, where seed rhizomes are stored in well ventilated place and covered with dry leaves so that it can be stored comfortably for 3-4 months. This is another method of storage where a layer of ginger rhizomes with the sawdust or sand, so that without any damage or shriveling, so rhizomes can be stored. And so this is again another method without any covering. So you can see the well ventilated, well shaded place. So ginger can be stored without any problem for 3-4 months. And again, as I said, it has to be treated again before the planting. The same like organic with the PGPR and inorganic with the recommended chemicals. And so this is the ideal planting method. So where we recommend like 25 to 30 gram seed rhizome is recommended with two viable buds. So that is the recommended seed size. So per acre, like you need average of 800 to 1000 kg seed rhizomes per acre. So so another important technology we developed, which I'm going to talk in detail, is transplanting technology in ginger, because you need like 800 to 1000 kg seed rhizomes per acre. So whether which can be reduced. So this particular technology is aimed to develop disease-free seed rhizomes. That is our major objective. Second, to reduce the quality planting material. So before going into details, I just would like to see this display this YouTube video where you can see uh, the highlights about this
sprout seed rhizomes into trays filled with soil nest medium.
Is it visible? Hello? Bhargav?
At the
No, sir. It's not visible, sir.
You
It's
have
not
to
visible?
buds with
It's
small
not visible, sir.
rhizomes swaying free.
Hello.
It's not visible, sir.
Okay, okay.
Hello.
Yeah.
So you have to share the screen on a YouTube actually.
Oh,
You
again.
can close the.
Oh, okay.
You have to click on that YouTube channel, and you have to, after this, you have to share that channel. Then it will be visible to us.
YouTube
You have to
channel.
close your PPT. You have to minimize your PPT. Then you have to share it again. That particular YouTube
Okay,
channel.
okay, okay. I thought
Particular
it.
window.
Oh, I thought it will be directly.
Yeah. Minimize it.
No.
Yeah. Minimize it, sir. Minimize it and share.
Once again, you have to
Okay,
click
okay.
the share screen.
Transplanting technique in ginger aids in the production of quality planting materials in a cost-effective manner.
No, it's not visible, sir.
It's not visible, sir.
Yeah.
Involves raising transplants from single sprout seed rhizomes into trays filled with soil-less medium.
Stop sharing, sir. Stop sharing first.
Now
it
it's
is
visible,
coming.
sir. Yeah.
Now it's coming.
Small piece of rice.
You have to play from the
Tray-based single sprout transplanting technique in ginger aids in the production of
Is it okay
quality
now?
planting materials in a cost-effective manner.
Yeah.
Yes,
Hello?
sir. Okay.
Yeah, okay.
Yes, sir. Okay, sir.
Okay, fine.
This unique technique involves raising transplants from single sprout seed rhizomes into trays filled with soil-less medium. At the commencement of season, pre-treated seed rhizomes are cut into single buds with small piece of rhizome weighing 3 to 5 grams. Before planting, the single bud sprout should be treated with 0.3% 9% for 30 minutes.
The pro trays are filled with partially decomposed coir pith and vermicompost in 3:1 ratio and should be enriched with plant growth promoting rice bacteria or trichoderma.
Ginger bud sprouts are then planted in pro trays, and the planted trays must be kept under shade-net house. Irrigation can be provided with rose canal by using suitable sprinklers. Seedlings will be ready within 30 to 40 days for transplant. The advantage of this method is the reduction in seed rhizome quantity and eventually reduced cost on seeds. This method also ensures the production of healthy, disease-free planting materials.
If the climate is not favorable for planting, the seedlings can be kept in shade-net houses for about 2 months.
Ginger rhizomes transplanted by this method shows 98 to 100% field establishment. In this method, the seed requirement is about 400 to 500 kilograms per hectare as compared to 2,000 kilograms in direct sowing.
Want to stand out online? Go to Wix.com and create a professional website that's sure to get you noticed. Start from scratch or choose from hundreds of people.
Hello.
Yes, sir. Now it's
Yeah.
visible, sir. Maximize the screen.
Yeah, so that video about this particular technology. So before going into the details, I just wanted you people to have a look. So this is the transplanting technology as explained in the video. So mostly to the produce the disease-free planting material. Then second is to reduce the initial seed requirement. So 800 to 1000 kg per acre, like 2000 to 2500 kg per hectare is a huge seed requirement. So definitely by this technology you can cut down the requirement to 250 kg per acre. So wherein you need only like 5 gram pieces, like it has to cut into small pieces of 5 to 7 grams with a viable bud. So each like piece should have one viable bud. Then like it has to be raised in the pro tray, so the seedlings will be ready within 30 days, and you can see 80 to 100% field establishment and saving in seed cost, high cost benefit ratio, and suitable for high production technology. And coming to the details of this particular technology, like it is like any other like vegetable seed production. So if you see now most of the vegetable crops, so the seedlings are produced in the pro tray or plug trays. So this is a 98 well pro tray is recommended for turmeric, and 50 well pro tray is recommended for ginger, because ginger we need slightly bigger sized wells. And the recommended potting mixture is the partially or well decomposed coir pith mixed with vermicompost and PGPR. So the ratio is 70% coir pith, 30% vermicompost or farmyard manure, whichever is available, and it should be enriched with trichoderma pseudomonas, like 10 grams per kg of the mixture. So this is the recommended mixture for ginger and turmeric. And another important aspect, I said, like we have to cut into small pieces. So you can have a closer look at this small pieces. Like you can observe each piece has a one viable bud. So without bud, like it won't germinate. The internodal region is cannot be planted. So only the nodal regions with a viable bud can be planted. So the recommended seed size is 5 to 7 grams, and you can see the viable seed piece. So and another area is like you can see uh, the recommended mixture filled plug trays. Uh, so another two, three practical observations here. So if I go back to this particular slide, so mostly what people do is like they cut ginger into small portions and they plant it in the pro tray. So the problems they face is there is no uniform seedling production. So that is mainly because so if you can see some of the buds are viable buds, so it's already started sprouting, and some of the buds are dormant buds. So if you plant all together, so one seedling will be 15-20 centimeter in height, so another will be just sprouting. So to overcome that problem, so what we recommend is uh, 10 days before cutting of rhizomes. So the seed rhizome, you can take it out from the storage, mix it with the coir pith, and you cover it with the polythene sheet. Like within 7-8 days, you can see the uniform sprouting of the all buds. Then you can cut into like small recommended pieces, then segregate. That plays the important role. You have to segregate according to the size of the sprouts, because this is mainly to get the uniform seedlings. If you segregate like the bigger buds in one tray, the smaller buds in another tray, the dormant buds in another tray, like the seedlings will be uniform. Otherwise, like you will have a problem of one seedling will be like very big, another will be just sprouting. So this kind of like pre-treatment of rhizomes for sprouting, then cut into small pieces, then segregate, and if you plant like 100% uniform seedlings will be produced. So another area you can see this picture. What generally farmers, they think like they cut like into small pieces, they fill with the coir pith, and this ginger pieces they put it on the top of the coir pith. So that what happens is like they think that the roots will come from the cut portion. So usually it is not. So it is not like vegetative cuttings. So usually the roots will emerge from the buds, the lower surface of the buds. So what care we need to take is the bud portion should touch the coir pith. So that it immediately strike the roots, and the roots will form like a ball in the coir pith. If you put it on the top of the coir pith, usually root won't strike, and so the piece is likely to be dried. So these two are the important the practical points you have to consider. So this is how the trays are filled, planted, and you can see like healthy seedlings within 25-30 days can be produced. And another, especially in the if your area is the temperature is very high, like you can have a small temporary shade net house, so that so it can be stored for 30-40 days. So you can see the turmeric produced in the similar way. So the ginger seedlings ready for transplanting, and you can see like once after 25-30 days, if you pull out the seedlings from the tray, so it should be like this. It should come out with the ball of the coir pith, intact coir pith with full of roots. Then if you plant seedlings like this, like you'll get 98 to 100% establishment. You can see another important and also the practical point to be considered is uh, like before planting, like the soil should be irrigated. So there should be a moisture before planting. So what people do South India is like they do the drip irrigation. They run the drip irrigation the previous day, so that the next day there is a sufficient moisture. Then they go for transplanting. So even in the case of rain-fed situations, if we don't have rains on that particular day, so we can wait for 2-3 days, so that there is a sufficient moisture in the soil for transplanting. So this is the transplanting under irrigated condition. You can see this is the transplanting under rain-fed conditions in Kerala. So you can see the sufficient moisture in the raised beds. So this is how it is transplanted. So this is in one of the farmers' plot in Karnataka, where it is transplanted under irrigated conditions. You can see the mini sprinklers all over. So after transplanting, they'll run the mini sprinklers, and you can see you can after planting you can do the mulching with the dry leaves. And this is one of the transplanted field of ginger in Karnataka. And this is the technology in short. So selection of healthy rhizomes, then seed treatment, as I explained earlier, then cut the buds into small pieces of 5 to 7 grams. Then you can go for one more seed treatment. Then 98 well pro tray for turmeric and 50 well pro tray for ginger. And this is the decomposed coir pith, vermicompost, and enriched with PGP are used as the potting mixture. Then planting of ginger buds, maintaining the pro-tray under shade net house. Then irrigation, like daily rose can irrigation. The seedlings will be ready by 35 days. And you can see this is the experiment we conducted, and we have conducted exhaustive trials on this. So T1 is direct planted. So T4 is transplanted, and T2, T3 are different sizes of rhizomes we used. And you can see T1 and T4, so there is no significant yield difference. So this is transplanted ginger. So initially there will be compared to direct planted ginger, so the transplanted will be slow for up to 75 days. So then thereafter, the growth will be uniform, and there is no significant difference in growth and yield of transplanted and direct planted ginger. So the main advantage, less of planting material, so only like 500 to 750 grams per kg per hectare, against like 2000 to 2500 kg of direct planting. So ultimately saving in seed cost, 100% establishment, high cost benefit ratio, suitable for high production technology. And like you can do, if you want early planting, you can go for 25 days. Seedlings can be ready for planting. Like if you don't have rain, so it can be delayed for 40-45 days also. So that flexibility is there with this technology. So to conclude this presentation, like we have good demand there is a good demand for quality ginger both in national and international market. And so we have technologies like varieties and quality planting material technologies to enhance the production. And another important area to be concentrated is product diversification. So this picture, like I have just put, like I can see, these are all some of the ginger products available in Australia. So Australia, so though they have only the small ginger growing area, but it is 100% processed. So they are the world leaders in processed gingers. You name any ginger product, ginger candies, ginger jam, jelly, or ginger beer, ginger based drinks. So they are the market leaders. So such kind of product diversification has to come into ginger, so that so that our farmers also will get benefit out of the ginger. So before closing this presentation, so I have one more small video on turmeric. So I'm sharing that video clip. So it's taking a little bit of time. Is it visible now? Hello? Hello?
Yes, sir. Yeah, it's visible, sir.
Yeah, this video about the transplanting technology in turmeric. So this particular technology, where students of the University of Horticulture Sciences, Bagalkot, So they have a college in Mysore. So where a student in their experimental learning program, so they are involved in producing the turmeric seedlings for distribution.
Steps to be followed in system of turmeric intensification. Healthy rhizomes are selected. Each rhizome is cut into small pieces weighing approximately 5 grams and should contain an eye bud.
You can see the rhizomes are cut into small pieces. So each piece has a viable bud.
Treated with solution containing phenol first 2 ml per liter.
So then they go for seed treatment.
For 10 minutes. Cut rhizome pieces are sown in grow trays containing 50 cells and each tray requires 2 kg of cocoa.
So you can see the cut rhizomes are planted in the media, so that the rhizome buds are in touch with the media. So even you can follow like this also. So after filling the trays, after planting, like you can cover it with polythene sheet for 3-4 days, so that the germination is uniform. So you can see how the seedlings are transported for the planting. So you can fold the trays like this, and so you can accommodate more number of trays. So nothing will happen to the ceilings. You can see just fold, and so that the more number of trays can be accommodated in a vehicle. So this particular video, like it's a student's work. So they have practiced this particular technology at College of Horticulture, Mysore.
Certainly, it will help us in bringing out the entrepreneur in us. We are thankful to all our teachers who guided us throughout this wonderful course.
So this is uh, so this is all about my first lecture. So like if you have any clarifications, we can discuss for 5-10 minutes. So
Sir, you continue with your second lecture, sir. Later, all together, we will take final, sir, at the end.
okay, fine. So just give me a minute. So I'll
Okay, sir. No problem.
so this second lecture is about the planting material production in black pepper. Is it visible now? Slides?
Yes, sir, it's visible.
Yeah. So so this is about varieties and planting material production in pepper. uh, so like I explained in ginger, so pepper also, like a few slides about statistics of pepper, so that you'll understand the importance of black pepper in India. So if you look at the India area and production, so it is cultivated in like 1,40,000 hectares with the 61,000 production. So mostly it is cultivated in Kerala, Karnataka, and Tamil Nadu. So now you look at the area of others, like almost 9,000 hectares of area like is cultivated in other states. That is mostly northeast. So northeast now, especially Assam and the bordering states of Assam, so where like winter is not severe, so it's highly suitable for black pepper. And especially in tea plantations, like now the pepper is coming up very well, and a lot of focus is now we are giving to extend the area of black pepper in northeast. So this is the just to give the overview of black pepper in the global level. So Vietnam is the major producer with 1,30,000 tons, followed by Indonesia and some of the like Southeast Asian countries. So the total production is 4,06,000 tons. So out of which Vietnam produces major junk, followed by Indonesia. So we produce around 60,000 tons. So, and also then the previous slide, if you see the productivity, the Vietnam, it is 2,300 kgs per hectare, dry pepper per hectare. Brazil, you see 2,000 kgs, Malaysia 1,500 kgs, China 1,000. It's all above 1,000. Even other countries, like it is about 2,000. But if you see the Indian productivity is only 300 tons. So before going to the reasons for low productivity in India, so the reasons for high productivity in other countries are like India. It is the pepper is cultivated as a component crop. So especially Karnataka, it is cultivated as an intercrop in coffee estates. Like in other states, like Northeast Assam, it is cultivated as intercrop in tea estates. So it's never cultivated as a monocrop in Indian system. So that is the one reason. So in the other countries, so like you can see the intensive cultivation system. So they grow pepper in the dead standards. So whereas we grow pepper in live standards. So that is the another reason. So they use wooden poles, concrete pillars, brick pillars. So the system of cultivation is very intense. So, and also the number of veins per acre. So under intensive cultivation system, so it's around 1,000 pepper vines per acre. So even if you take like 1 kg also, it is 1,000 kg dry pepper per acre. Whereas our system as a component crop, so we have only around like 50 to 100 vines per acre under coffee and tea plantations. And another important point is in the like countries like Vietnam, Indonesia, Malaysia. So they have a bimodal distribution of rainfall. It means like throughout the year they get rainfall distribution. So that is very conducive for pepper production. So they get to 2 peaks in a year, whereas in Indian system, so we get only 1 peak. So monsoon starts from June, maybe August, September. August it will be peak, then by November it will be over. So only 1 peak. The rest 5 months it will be a dry period. So this is the pepper plot in Cambodia under intensive system. And coming to the reasons for low productivity, in India, it is a poor genetic potential of the vines, high population of old and unproductive vines, and the major problem of diseases, especially the Phytophthora foot rot, and major our emphasis here is non-availability of quality planting material production. So that is why we are here for this lecture. And the major like biotic and abiotic stresses, disease, Phytophthora foot rot, major problem. Again, nematodes, Meladogine, Rhadophilus, major problems. Like to overcome that, like you need to produce the quality planting material, disease-free planting material. So coming to the varieties available in black pepper. So like ginger, the pepper, the diversity is very high. The crop is originated in South India. So the diversity is huge. So there are 38 common cultivars. Besides that, there are around 60 less important cultivars. So there are around 110 cultivars are available in black pepper. You can see some of the cultivars like Kalluvalli, drought tolerant, even. So this particular cultivar, Karimunda, like with short spike, bold berries, and there are many local cultivars with the diverse characters. So from the Indian Institute of Spices Research, so we have released 8 varieties: Subakara, Shrikara, Panchami, Paurnami, PLD2, or some of our old varieties. And like 10 years back, we have released IASR Tevam. Then like we have 2 hybrids, Malabar XL, Girimunda, then IASR Sakti, which is tolerant to foot rot. So these two are one of our old varieties, Srikara and Subakara, which are selections from local cultivar called Karimunda. So the advantage of these two varieties are high essential oil content varieties. We can see essential oil of 6 and 7%. So besides that, consistent yielders, like varieties suitable for sustainable production. Like if you do intensive operation, like if there is a rain, there is a no rain, there is a high shade, low shade. So these varieties will perform consistently. So these two varieties are suitable for sustainable production. You can see this Shubhakara varieties, you can see number of spikes and the number of leaves. This we call it as sink source relationship. So number of leaves are very less, whereas the number of spikes are very high. So this is very desirable character for black pepper. And this is another variety released from Indian Institute of Spices Research called PLD2 or Pollowed 2. The important character of this variety is high oleoresin content. So generally oleoresin content in pepper is around 10 to 12%, where you can see this particular variety has 15.5% suitable for oleoresin extraction industries. And Panchami, Paurnami are another 2 varieties. Paurnami, which is tolerant to nematodes, and Panchami is a selection from Einberian, and which is suitable for like areas with the high shade. So this particular variety is suitable. And you can see Paurnami, which is tolerant to nematode, and this is Panchami, suitable for high shaded areas. And ISR Tevam is tolerant to Phytophthora. Then ISR Sakti is also tolerant to Phytophthora. So these two varieties, especially where you have the problem of the foot rot disease, so we can recommend these two varieties. So you can see the performance of ISR Tevam in one of the farmers' plot in Karnataka. You can see the spike and leaf characters of ISR Tevam. Then we have two hybrids, ISR Grimunda and Malabar Excel, which are suitable for high elevations, like more than 1,000 meters MSL. So these two hybrids are suitable for higher elevation. You can see the intensity of spikes in Malabar Excel. This is Grimunda, and again Grimunda in one of the farmers' plot, where it is uniform in maturity. Then besides that, Indian Institute of Spices Research and IIHR Bangalore, so collaborative through our collaborative efforts, so we have released a variety called Arka Kurg Excel. So this particular variety is recommended for Karnataka. Besides that, Kerala Agriculture University, so they have a pepper research station in Pannur. So they have released not 7, so they have released 9 varieties as on date, Pannur 1 to 9, out of which Pannur 1 is Pannur 1 and Pannur 3 are hybrids. Pannur 1 is quite popular variety in pepper. You can see the performance of Pannur 1. So very long spikes with bold berries, and the variety is highly suitable for intensive cultivation system. So it means it's highly input responsive. So if you apply more fertilizers, so it responds to that and it gives more yield. You can see in the Pannur 1 with integrated nutrient management, you can see the number of spikes in a square meter. It's very high, very good variety. And another variety like Pannur 4 has wider adaptability than Pannur 5, suitable for shaded areas. Pannur 7 with long spikes. Pannur 8 tolerant to Phytophthora and drought. So wherever you have the problem of foot rot disease, so we can recommend Pannur 8. And this is the Karimunda. Many of our selections has come from this particular local type Karimunda. And Kottanadan, again a local type which has high the oleoresin content. Like another consistent local cultivar, Narayakudi. Then this is a particular local cultivar called Chomala, so which is cultivated in tea estates in Tamil Nadu and Kerala. So suitable for the open areas. So you can see the berry characters. The first, this one is Pannur 1. So this is another local type called Vadakan. The berries are very bold. So the Karimunda, which has small berries. So, so, so yield potential, like you can classify like pepper varieties like this. So yield potential like Pannur 1. So if you want varieties for resistant to Phytophthora, ISR Sakti, Devam, and Pannur 8. Then varieties resistant to nematodes, Paurnami, resistant to moisture stress, Pannur 6 and Pannur 8. Then varieties suitable for high elevations, Malabar Excel and Girimunda. Varieties suitable for intercropping in coffee estates, Pannur 1 and 5. Then varieties suitable for high quality, bold berries, shade tolerant varieties, and varieties with high dry recovery. So coming to the planting material in production, like as I said, Phytophthora and nematode are the two major production constraints in black pepper. So again, like there are chances nematodes and this Phytophthora can be carried away along with the planting material production from one state to other. So planting material plays a major role where we need to supply the disease-free, high-quality planting material production. So here the requirements for quality planting material or mother plant. So that is very important. So the joinality and maturity, whether you can take cuttings from young plant or whether you can take cuttings from old plants. So like generally what we recommend 7 to 15 years old mother plants are suitable for planting material. And genotype, like you have to select the true to type, like genotype for maintaining the purity. Then nursery media, plant growth substances plays important role, and then propagation structures. So coming to the potting mixture, especially like this Phytophthora and nematodes, which can like multiply in the nursery. So we need to take extra care in potting mixture. So generally the potting mixture, these three are important points. Like, so there should not be any excess water in the potting mixture. So it should be porous in nature. So that is the first point. Second point, like it should have adequate the balanced nutrients in the potting mixture. Then it should be free from harmful pathogens and nematodes. So these three are very important for pepper. So the ideal potting mixture usually follow is 2:1:1 soil, sand, and farmyard manure. Then we recommend soil solarization so that the pathogens and the nematodes can be eliminated. So then after solarization, you can enrich the potting mixture with trichoderma. So in the you can also use like coir pith and vermicompost as a soil-free medium in the case of advanced nurseries. Then even if you have the problem of getting the sand, like we have a substitute like granite powder can be substituted for sand. Then azospirillum and phosphobacteria can help in better rooting. And this is how the potting mixture is to be solarized. Uh, this is the soil solarization of the potting mixture. It is very, very important. So I have a YouTube video on solarization. Is video visible?
A well-prepared nursery mixture
Hello?
will provide good nutrition.
Yes, sir, it's visible.
Okay.
Yeah. Yes, sir, it's visible.
To prepare the nursery mixture for planting the cuttings, take 2 portions of soil. Topsoil is the best, sand and manure, and mix them thoroughly. If possible, this mixture should be solarized to get rid some of the soil-borne plant pathogens. To solarize, the soil mixture is moistened and spread on a level surface with 1 meter width, 25 centimeter height, and of convenient length. These beds should be located in an area exposed to bright sunlight. The beds are covered with a transparent polythene sheet of 100 to 150 gauge without leaving any air gaps. The beds are to be sealed on all the sides with a mud plaster. This would ensure heat buildup and initiate a process called solarization. You can use the solarized nursery mixture after 35 to 45 days. To prevent pathogen contamination, the solarized nursery mixture should be immediately mixed with biocontrol agents such as Trichoderma hertzianum or T. viridae, 1 gram per kilogram of nursery mixture, Pseudomonas fluorescens, strain P1, 20 grams dissolved in 1 liter water, and 50 ml of this solution to be used for 1 kilogram of nursery mixture. WAM or AMF, vesicular arbuscular mycorrhiza or arbuscular mycorrhizal fungi, at the rate of 100 cc per kilogram of nursery mixture, and Pochonia chlamydosporia 1 to 2 grams per kilogram of nursery mixture. Such solarized nursery mixture mixed with above biocontrol agents would prevent root infection in the pepper cuttings. When solarization of nursery mixture is not possible, unsolarized nursery mixture can also be mixed with biocontrol agents that would protect roots from infection. The nursery mixture is then filled in poly bags of size 20 into 10 centimeter or 15 into 10 centimeter with enough holes to ensure proper drainage. Please download the IPC Farmers app for your country from the Google Play Store or from the App Store for more training videos. Guidance on cultivation, daily price information, marketing support, exchanging ideas with other farmers.
So the video what you have just seen is about soil solarization. So that is very, very important, especially for black pepper nursery. So coming to the second important aspect, selection of mother plants and the shoots. So you need to understand this particular point for black pepper nursery. So there are three general shoots are available in pepper. So one is runners. So you might have studied in your the basic courses in spices and plantation crops. So one is runner. So the mainly it is available in the base of the plant, so which is used as the planting material. So the second, the orthotropic shoots or top shoots. So which is mostly in the top erect growing shoots. So which is again you can use it for planting material. So the third type of shoots are plagiotropic shoots. So which are the usually the bearing branches. So wherever the spikes are there, so they are called the side branches or laterals or plagiotropes. So these plagiotropes are not used for propagation. So if you plant these plagiotrops, so it will not grow as a vein. So it will be maintained as a bush pepper. So usually these plagiotrops are only used for bush pepper production, whereas runners and orthotrops are used for commercial nurseries. So ideal characteristics of mother plants: you avoid young plants up to 7 years of age, avoid old plants too. So some of the 40-50-year-old plants also not suitable for mother plants. So ideally, like usually like 7 to 15-year-old plants are recommended for taking runners and top shoots for multiplication. So even you can see some of the planters, what they do. So the runners, so they simply coil and put it on the poles. So this is mainly because to avoid the soil-borne pathogen infection. So immediately after the monsoon. So before taking the cuttings, 2-3 months before, so they will lift the runners and put it on the poles so that it will not take any further infection. So this is how the terminal shoots are used for propagation. Especially these terminal shoots are used in Southeast Asian countries, especially Vietnam, Indonesia, where the terminal shoots will come to bearing early, maybe compared to runners. So this is like 1 or 2 years early in bearing, and also like the bearing habit is also quite distinct from runners. So it starts bearing from the like from the ground level itself, whereas like you can see when you plant runners from the ground level up to like maybe 1 meter, so there is a gap. So after that only it starts bearing, whereas the top shoots will start bearing from the ground level, and you can see the performance of plants derived from the terminal shoots. And like propagation, like we propagation methods are single node cuttings. So cuttings or single node or two node cuttings are used, and we have a rapid multiplication technique, either serpentine method or rapid multiplication technique. Then we have the new technology based on soilless media, where we use plug trays for producing disease-free planting material. So this is the single node, two node cutting nursery. You can see either you can use single node or two node cuttings. Usually this is taken from the runner shoots. So it is planted in polythene bags. Then to have a better rooting, so these kind of small tunnels covered with polythene sheets, so that humidity is maintained inside, so that the percentage of success is high. Usually this polythene covering is given for 60 days, and like after that like you can put it for hardening for another 60 days. So the plants will be ready for planting at the 3 to 4 leaf stage. So here the single node or two node cuttings, the success rate is around 70 to 90%. You can see the polytunnels. So immediately after planting, you can put a polytunnels, maybe once in 2 days. So you can open and water, and again you cover it, so that the humidity helps better rooting. You can see the picture given down. You can see the just sprouted. Maybe 50 to 60 days, you can slowly remove the polythene sheets. So so this how the sprouting comes from the single node cuttings. You can see this is after removing the polythene sheet, so you can shift them to the hardening shed. So you can see the uniform single node cuttings produced. So so another the problem uh, single node cuttings, like it may have sprouts but it may not have roots. So that aspect we have to take care. uh, like you can see this single node cuttings has sprouts, but it doesn't have the roots. So the proper maintenance of the nursery is very, very important for proper rooting of the cuttings. And the second important this one is rapid multiplication. This is the bamboo maced bamboo method of rapid multiplication, where you can put the split bamboos filled with coir pith based media, and you can plant the pepper cuttings at the base of the bamboo. So within 45-60 days it grows to the top, and it strike roots in the bamboo filled with coir pith. Then after 45-60 days, you can remove the entire rooted cuttings. So cut into individual nodes and plant it in the polythene bags. So here the multiplication rate is 1:40. So if you plant one cutting in a split bamboo, so in a year you can multiply 40 cuttings. So the success rate is 90 to 100%. So this is how the technology, the split poles of bamboo are fixed at 45-degree angle, filled with rooting medium, the mostly coir pith based medium. Then once it reaches the top, then you have to remove the entire vein with the 10 to 15 nodes rooted. Then you can plant it in the polythene bag. So you can see this is again the rapid multiplication followed. I think this picture we have taken in Assam. So this is amalgamated coffee plantations. So based on our advice, so they have a huge nursery for multiplying IASR variety, IASR Tevam in their nurseries. And now the entire amalgamated plantations of Northeast Assam, part of Nagaland, Manipur. So they have this particular variety in their plantations. Uh, it's a Tata Group estate, and so this is another modification. So it's a farmer innovation where instead of bamboo split, so this particular farmer uses 5-inch PVC split into two. So it holds like more of coir pith. So instead of one particular vein, like you can put 2-3 veins so that you can harvest more number of cuttings. And you can see the rooting of cuttings in the rapid multiplication technique. So another important method is serpentine method. So in the case of rapid multiplication, again you have to cut into individual nodes, and it has to be planted in polythene bags. So here in the case of serpentine method, you can see this particular picture. So like when it growing, you keep the polythene bag with the media at the each node, so that you need not again replant in the polythene bag. So it strike roots readily in the polythene bag, and you can see. So as and when it grows, like you have to keep the polythene bag filled with the media to the each node. Then so after 15-20 nodes of growth, so you can leaving top 5 nodes. So the rest of the nodes you can simply detach it so that it will be independent. Yes, it already strike the roots. So serpentine method, like advantages, like bamboo splits are not required. So replanting is not necessary. So multiplication is 1:60 compared to bamboo method where it is 1:40. So this is the serpentine method. So again, I'm not sure whether you people have sufficient time. I have one more video on serpentine method.
Rooted cuttings, serpentine method. As explained in our training video on how to prepare rooted cuttings, prepare the rooted cuttings in 20 into 10 centimeter poly bags. Select rooted cuttings that have grown well and have 3 to 4 leaves. Arrange the selected cuttings at one end of the nursery, and each cutting is trained to grow horizontally. As shoot grows, bags filled with nursery mixture are kept underneath each node and allowed to strike roots. It is essential to ensure that each node touches the mixture. The shoot is then allowed to grow for about 3 months so that it can produce about 10 to 15 single-noded rooted cuttings in a batch. This process continues in such a way that a single node gets rooted in each bag until the shoot reaches the last bag in a row. Once all nodes get rooted, the first 10 polythene bags with rooted nodes should be separated by cutting the internodes. Fill the poly bag with more soil to ensure that the cut ends are below the soil surface. The axillary bud from each node in the bag sprouts and develops into 4 or 5 noded rooted cuttings in about 2 to 3 months. After a gap of another 20 to 30 days, the rest of the bags also can be separated. About 40 to 60 rooted cuttings can be produced from a single plant during a year. Please download the IPC Farmers app for your country from the Google Play Store or from the App Store for more training videos. Guidance on cultivation. Daily price information.
Hello.
Yeah, hello, sir.
Yeah.
Visible, sir, slides.
Yeah, so the video what you have seen is about serpentine method. So this is how the serpentine method is followed in our IAS or nursery, and you can see the cuttings produced from serpentine method. So and like we have like a modified serpentine method. So wherein you need not keep the bags at the each node. Instead of what you can do is you can make a coir pith based bed. So wherein you can trail the pepper runners on that. You can see. So this is just a coir pith based beds. So you can trail the pepper runners on that. So after that, once it grows 15 to 20 nodes, so you can cut into small individual nodes, and you can plant it in the nursery bags. So you can see the coir pith beds. Uh, so once it reaches 1.5 meter with 10-15 nodes, so it can be detached individual node wise and planted. You can see you can detach so that it sprouts. Then you can plant it in the polythene bags. So this is how we have developed technology, wherein like initially it is modified serpentine method, where it is strike the roots in the coir pith based media. So then later it transferred to the plug trays. So where the slightly the bigger plugs are used, the same coir pith media composition 75 25 like a coir pith vermicompost ratio enriched with PGPR. So 45-60 days you can see the beautiful sprouts. Then again 45 to 60 days hardening. So the 120 days, like you can get the beautiful plugs like any other horticulture crops. So 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 that the main advantage of this technology is like the movement of the planting material, especially the black pepper. What happens is like if you supply the black pepper cuttings in the polythene bag with the soil, what happens is during the transportation, like because of the heaviness of the soil and the very delicate nature of the black pepper roots, it get detached. So the establishment is very less. So to overcome that, like if you supply in the plugs, so it is very convenient for transport, and like establishment rate is also high. And again, the advantage, like it is soilless media, so the you can overcome the problem of diseases here. And you can see the while planting, it comes out as a plug of coir pith ball, so that if you plant, there is 100% establishment. So another method, like you have seen the modified serpentine method with the coir with bed. So you can also like make a vertical method, like you can make a iron mesh based poles with the coir with mixture. So you can allow the black pepper plants to trail on this. Here the multiplication ratio is quite high. So even compared to the beds. So once it reaches the top, so you pull out with the roots and plant it in the plug trays. So you can see the rooting at the individual nodes. So then later, you can trim it and you can put it in the plug trays. So this is the technology for disease-free planting material in black pepper. You can see serpentine method, or like in the greenhouse, you can grow it, then put it in the plug trays. 45-60 days it roots beautifully with sprouts. Then another 45 days for hardening. So the plants are ready for planting with 120 to 150 days. So that is the cycle we follow. And so I have this is my last video, so where this video is for this plug tray production.
Column method to produce black pepper cuttings. Black pepper, the king of spices, is one of the oldest and the most popular spice in the world.
This perennial vine is mostly grown for its berries, which are extensively used as spice and medicine. Generally, stem cuttings are used for its propagation.
Although many improved pepper lines have been evolved, insufficiency of quality planting materials and incidence of soil-borne diseases are leading problems confronting the growth and development of pepper. The earlier methods enable making only single or double node cuttings and do not provide top shoot or lateral shoot. Column method is a novel, 3-in-1 method for large-scale production of quality planting materials of black pepper, including top shoots, lateral shoots, and normal single-node cuttings.
This technique utilizes soil-free medium, ensuring the production of healthy and pathogen-free cuttings within a short period of time. Vines from these cuttings give better growth and earlier flowering, which enable the plants to be more productive.
The technique involves growing the rooted cuttings on a welded vertical wire mesh column of about 2 meters height and 30 centimeters diameter. The column is packed with partially decomposed coir pith and vermicompost in 3:1 ratio and enriched with trichoderma, or plant growth promoting rhizobacteria.
8 to 10 uniform polybag cuttings of black pepper kept around each vertical column are then allowed to trail on the column.
The columns with black pepper cuttings are maintained in a high-tech polyhouse with controlled temperature and humidity.
As and when the cuttings grow, medium is filled in the column.
Nodes are firmly fixed along the column by means of coconut leaf splinters. This allows the roots at each node penetrate through the medium in the column and develop as functional root.
Once the vines have reached the height of the column, top shoots with 5 nodes are harvested and kept on pull trays for further development.
Cuttings from lateral branches are used for healthy bush pepper production. Remaining nodes are separated as single nodes and planted in trays for raising normal cuttings.
This highly productive method can yield about 30 to 40 top shoots, 50 to 60 lateral shoots, and 250 to 300 normal shoots from a single column in about 1 year with 3 harvests.
These normal cuttings are suitable for planting as intercropping coffee and tea plantations, since the cuttings are capable of developing lateral branches after a height of 2 meters. For pure plantations, the productive shoots must start from the base, and hence top shoots are ideal for this.
Also, the top shoots when planted bear spikes within a year as against 2 to 3 years in conventional method.
The vertical method ensures the production of top shoots as well as lateral shoots as addition compared to horizontal methods.
This method has the power to meet the bush pippa planting material and top shoot requirements of farmer.
Hello, am I audible?
Yes, sir, you are audible.
Yeah. So last 5 minutes of my presentation, so there are some modifications of like what we so far what we have seen. So this is a particular method where the one farmer in Karnataka, so he follows, so instead of collecting runners and planting, so what he does is he puts the polythene bag filled with the potting mixture in the plant itself, so that he gets the cuttings, rooted cuttings readily, so it can be planted directly into the field instead of taking single node and double node cuttings, then rooting and planting. So he directly roots them like this and go for the planting. And so this is the another method of air layering, so which can be followed in pepper, especially for top shoots. So you can put a air layer, so after rooting then it can be directly planted. So this is the grafted pepper, so which is followed in some places in Karnataka where water stagnation is the major problem. So here the rootstock is a different species of piper. So you might have heard about the piper colubrinum, so which is suitable for waterlogging conditions and tolerant to foot rot and phytophthora. So here Piper colubrinum is used as a rootstock, and the cultivated Piper nigrum is used as a scion. High yielding varieties are used as a scion. So, and it is quite successfully followed in Karnataka, where the water stagnation is a problem. So these are all the grafted plants of Panure 1, grafted on Piper colubrinum. So this is in short about the quality planting material production of spices. So especially ginger, turmeric, and black pepper. So which are the these three crops are the focus crops for the Northeast. So I hope you people get benefited by this presentation. And if you have any specific queries, so I am happy to discuss for a few minutes. Thank you.
Yes, sir. Thank you very much for your nice presentation, sir. Sir,
Okay.
now we can have some questions, sir, in chat box.
Okay.
Where we can get the quality ginger and turmeric seed material. Kindly provide contact number.
What is that?
Where we will get the quality ginger and turmeric seed material, kindly provide the contact details.
No, the ginger and turmeric, like you can get it from Indian Institute of Spices Research. So you can write or you can email to director uh, uh, spices at icr.gov.in. So it's available in our website www, like Indian Institute of Spices Research website. If you go, you have complete contact details. So otherwise you email to me, I'll be happy to share the contact details.
Okay, sir.
Yeah.
Next question is how much it costs one tray of turmeric or ginger seedlings, one complete tray, how much it costs?
No, it depends on like size of the pro tray. So what we are targeting is like it should be like 1 rupee per seedling. Okay. So either ginger or turmeric. So the production cost will be around 75 paise. So the entrepreneurs or the farmers who are producing, no, he can sell it for 1 rupee per seedling. So he'll get like 25 paise margin. So even if you sell for 1 rupees also, it is economical to the farmers also. So the production cost will be around 75 paise. So the nursery people can sell it for 1 rupee.
Sir, how many plants are required to plant 1 acre of ginger or turmeric?
In the case of ginger, it is around 15,000 to 20,000 plants per acre. Okay. So yeah, 15,000 to 20,000 plants per acre. In the case of turmeric, it is 28,000 to 33,000 plants per acre. So it depends on like spacing. So I'm telling the general range like 28,000 to 33,000. No, so some of the short duration varieties, like you can plant more number of plants, so that plant population is 33,000. So some of the vigorous long duration turmeric varieties, so you can have only 28,000 plants per acre.
How best we can store turmeric on field for next season? What is the best and economical method of storage?
Yeah, you are right. Like provided, like if your area is seeded and there is no like temperature issues and there is no direct hot sun, so it can be stored in the soil for 3-4 months. There is no problem. So only issue comes like if it is rains in between, then all ginger will be start sprouting. So that is the another issue we have to take care. Anyway, it is we cannot help that one. If it rains in between, like within 7 to 10 days, it will be start sprouting. Hello.
Now you can. I'll unmute all of you. You can ask directly if you have any question. I'll just allow you to unmute.
Good afternoon, sir. Good afternoon, sir.
Good afternoon.
Good afternoon.
Yeah.
Good afternoon. Yeah, audible?
Yeah, audible. Yes.
Sir, my name is Tejram Nagar. I am grower of ginger in 20 acre.
Okay.
And nice presentation on ginger and turmeric. A lot of thanks, sir. You giving very advanced technology and knowledge for better farming. And कहना चाहिए इससे हम काफी सारा पैसा भी बचा सकते हैं. तो मेरा ये सवाल है कि इससे सर कितना प्रोडक्शन इंक्रीज होगा? हाउ मच प्रोडक्शन इंक्रीज कंपेयर विथ ट्रेडिशनल जिंजर एंड टर्मरिक फार्मिंग व्हेन वी यूज Seedlings preparation of ginger and turmeric.
No, you are cultivating ginger, right?
Yes.
So what is the average yield you are getting in ginger?
Approximately 8 to 10 ton per acre.
8 to 10 tons fresh ginger per acre, right?
Per acre, 8 to 10 ton. Yes.
Which area? Where is your farm?
MP, Ujjain, Madhya Pradesh, Ujjain.
Oh, Madhya Pradesh Ujjain. Okay, fine, very good. So now, if you go for transplanting,
Yes.
so whatever the technologies you followed for your other ginger, no, so the same technologies you can follow for transplanted crop also. So the yield will be same. So whatever you are getting in the traditional crop and the transplanted crop, so the yield will be same. So there is no reduction in the yield. So that's what we are trying to say by transplanting technology. Okay.
Yeah.
So
Yeah. Yeah.
so only advantage for you is like if you want to cultivate 20 acres of ginger in Madhya Pradesh, like you need 20 tons of ginger seed material, right?
Yeah. 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 Yeah.
Yeah. So if you want to raise the turmeric seedlings, like you need only like 5 tons of the seed material.
Yeah. 4 time seedlings requirement is less compared
Yeah,
with traditional method.
exactly. Your seed requirement is very less, so you can cut down your seed cost.
Yes. Yes. Yes.
Yeah,
Yes.
yeah.
And early maturity compare with traditional method. 1
Early
year, 1 month, 1 month.
Yeah, 1 month. Yes, yes.
Sir, can you share your contact number with me?
Yeah, yeah, no problem. So you can note down my mobile
Yeah.
number is 9495
9495
63
63
9838.
9838.
Yes.
Because very good work, sir, on your ginger and turmeric. I very impressed. Thank you, sir.
Oh, thank you, thank you.
Ji.
So you can contact our institute like next year even so we can take up some trials in your farm. Next year we'll have
Yes,
some trials there in your farm.
yes, yes, sir. Yes, sir. I interested. I interested.
Okay.
Very interested.
Okay, thank you.
So any other participant? Would you like to ask any question? I think um, so. Really, I could hear your presentation from the beginning to the end, sir. It was very nicely meticulated way you have prepared the presentation. The PPT was very nice, and you have thrown a lot of light for the ginger planting techniques. It was very interesting, like raising, in the nursery and single cuttings. It was very nice. I would like to ask one question, sir.
Yes, sir.
So yeah, this northeast disease is a major problem.
Yes, sir.
So disease that is like rhizome rot along with the nematode is a more problematic. Is there any resistant variety for Pythium or Fusarium resistant varieties for nematode also? If you have, you can please share with us.
Yeah, you have raised a very relevant question, sir. So as on today, to be frank, there is no resistant variety either for Pythium or Ralstonia in ginger. So because the ginger conventional breeding is not possible in ginger because ginger doesn't set the seed. So there is no possibility of doing the conventional breeding. So that's why the variability is very less, especially against diseases, and there is no resistant source available within cultivated ginger. So here in Indian Institute of Spices Research, so last 5 years, we are working on mutation breeding in ginger, especially to induce variability against diseases. And so we have shortlisted a few promising mutants. So these mutants are under multi-location trials under All India Coordinated Research Project on Spices. So maybe like in a year, in a 2 year time, so we have every chance to come up with a tolerant variety against Pythium and Ralstonia, sir.
Sir, one more question. Like in one of your slide, you have shown that the raising of ginger on month, like without any shade. I could not see they have not any planted any shade crop. Like here, they used to plant this red gram, and or else in the forest, virgin forest, they'll cut off, and then in a zoom cultivation, they'll allow some trees to provide shade for the ginger. So there is a huge yield. But I could not see in some of your slide that there is no shade. So here, if they are not providing proper shade, there is a problem of cercospora leaf spot on ginger.
Yeah.
So what is the reason you are not recommended any shade plant for that ginger which is blown? Do you have any reason, or will they attack by any? Are they free from disease? Or I want to comment on that slide.
Yeah, sir. Here in South also, like earlier, like 15 years back, ginger is cultivated in like subtropical conditions, especially in Kerala, like Wayanad and Idiki conditions. So now what happens is like the ginger farmers slowly they are spreading, they are going out of traditional areas to non-traditional areas. So the non-traditional areas in Karnataka, it's quite a dry area, like you pointed out correctly. So it's a dry area, so not much rainfall. So the advantage is the area is free from diseases. So when you have high rainfall, so you are likely to be with more of diseases. So the dry areas, the first thing is very less diseases. So they are able to manage the dry conditions with the help of mini sprinklers. So I was pointing out in one of the pictures. So every 10, 12 feet, so they have a mini sprinklers. So that they create that microclimate, so that every day, like evening, like 4 to 6 or 4 to 7, depending on the dry conditions, they operate the mini sprinklers, so that the humidity is maintained, that the microclimate is maintained. That is second thing. So the third point is the genotypes, especially the Rio de Janeiro, which is cultivated in South India. So it is relatively free from the Philosticta leaf spot. So only they prefer this particular variety for this open area. So whereas the other genotypes, especially if you open up the shade or you put the ginger in the open conditions, immediately it will be susceptible to Philosticta leaf spot. So the open areas, they prefer this particular genotype Rio-de, which is comparatively like tolerant to this Philosticta leaf spot.
Yeah. Thank you so much, sir.
Yeah.
And participant, is there any question? Okay. Yeah. Oh, they were asking one question, like how much production of ginger and turmeric per acre? There was a question.
Yeah, so especially ginger, that's what that farmer from Madhya Pradesh, he is telling that he is getting around 10 tons per acre fresh ginger. But under intensive cultivation system in Karnataka, where that particular photograph is taken with mini sprinklers, and with many rounds of fertilizer application, pesticide application. So the yield level can go up to 25-30 tons per acre. So that is the yield level in ginger. In the case of turmeric, again it varies from 20 tons fresh turmeric per acre to 30 tons fresh turmeric per acre. Especially in if you go to Tamil Nadu, especially Rio-de, the Gobi Chettipalayam belt where the turmeric is cultivated, so they take around 25 tons fresh turmeric per acre. So Northeast, like people take around 10 tons per acre. The productivity level is around 10 tons.
Yeah, the productivity is very high, sir. I have visited many of the places.
Yes.
Yeah. Thank you so much, sir. It was more informative and enlightening to all our participants. I think afternoon session will be very difficult, but you made it alive, sir. Everybody was very active.
I hope so.
When
I hope so, sir.
we could even I had a lot of other work or Dean sir was calling, but I could not go because your slide was so interesting. I was waiting to finish you, and then I will go for the
Oh,
go
thank
to
you,
the
sir.
office.
Thank you very much.
So it
Thank
was
you
very
very much.
informative, sir. You have provided a lot of knowledge on the ginger, turmeric, and pepper and other crops propagations, and it is
Yes.
a very interesting thing for our participants and students. We are conducting one competition, students innovative idea competition for the students. The first
Okay.
winner will be getting 10,000, and second prize will be 7 and 5,000.
Oh,
I
very
think
good.
students, if they are, I think from you they can take a lot of ideas, and the way you have presented, I will not reveal it, but then dear participant and my dear students, you can go to once again sit back and watch his entire video, and you can get a lot of innovative idea for the northeastern region as per our team area. If you could take out and present it, you can win 10,000 rupees. Okay, so last date is 10th November. Be ready for that. Okay, so really we are from our College of Horticulture and Forestry on behalf of IDP Nahe project. I would like to thank you very much, sir, for your nice presentation, for sparing your time. And before closing up, I would like to ask Taso Yatung, Assistant Professor, would like to propose the vote of thanks.
Yes.
Thank you, sir, for giving me the opportunity. Okay, good evening, one and all. I welcome everyone to the last part of the day. So to begin with, I would like to thank Dr. Basanta Singh, sir, Director of Instructions in Agriculture University, Professor B.N. Hazarika, Dean and Nodal Officer, IGP Nahe, College of Horticulture and Forestry, Tekul Raja, Associate Nodal Officer, IGP Nahe, and our very learned speaker for the today's program, Dr. A.K. Dubey, sir, Principal Scientist, Division of Fruit Science, IRI, New Delhi, Dr. A.K. Singh, sir, Principal Scientist, Division of Vegetable Science, IIR, New Delhi, Dr. D.P.S. Raju, sir, Principal Scientist, ICR, Director of Floricultural Research, Andhra Pradesh, Dr. D. Prashanta, sir, Principal Scientist, Division of Crop Improvement, ICR, IISR, Kozhikode, and Dr. Ramakrishnan, Managing Director, Sri Satya Deva Nursery, Kadiyam, Andhra Pradesh, for sharing their knowledge and experience. I would also like to thank our dear participants who, in spite of yesterday being holiday, were with us till today. Like there was a gap of one day, but they attended the session. And lastly, my thanks goes to the organizing secretary, Dr. Velaru Bhargav, who took the pain of bringing all the learned persons to a single platform for our young entrepreneurs, encouraged them to take up nursery as a business venture. Last but not the least, I would like to thank all the committee members and people who worked behind the scene to make this program a success. Thank you once again. Thank you.
Yeah, thank you, Taso, for proposing the vote of thanks. With this, we are coming to an end. Before end the Zoom meeting, I would like to request every participant.
Can I ask you one thing? Sir, can you hear me? Sir, can you hear me?
Hello.
Sir, can you hear me?
You are asking me, sir, or hello?
Yes, sir, to the organizer, yes.
Oh, organizer. Okay.
So everyone, please start your video. I request everyone to start your video so that it will be record of your attendance and so on. Please start your video, everyone.
Myself, Sandeep Nath from Tripura, sir.
Yeah, thank you so much. Sir, with this, I'll end the meeting. Actually, yeah. With this, we will end. Thank you very much. Thank you, sir.
Thank you to all.
Yeah, thank you very much.
Thank you.
Thank you.